But before beginning our topic, I would like to remind you all about our Unacademy Plus subscription, which is very, very beneficial for all the aspirant EAMCET. It is a premium prices, okay? At affordable structured life courses plus is there. Then we have quizzes at regular intervals for self-analysis. Quizzes and batches are there for complete preparations. There are test series for DPPs, full-length mocks, etc. with organisms living everywhere. Yes, bacteria. Now, what exactly is bacteria? Bacteria, they are the most abundant organism on the earth. They are the widely present everywhere. Okay. Yes, bacteria are the most abundant organism on earth. And Anton von Leeuwenhoek first discovered this bacteria. Who has discovered bacteria for the first time? The famous Anton von Leeuwenhoek. He was the first one to observe bacteria, and later on Ehrenberg coined the term as bacteria. Ehrenberg has to coin and gave the term as bacteria. Okay. Then Louis Pasteur is the father of bacteriology. Louis Pasteur is the father of bacteriology. Why he is considered as father of bacteriology? Due to his extensive work. Due to his extensive work, and he showed that bacteria is capable of bringing significant changes in nature. Who said that bacteria is capable of bringing significant changes in nature? It is Louis Pasteur, and hence due to his extensive studies on bacteria, he is considered as father of bacteriology. Yeah, then these can withstand extreme cold. They can withstand extreme cold, heat, and drought condition. They can withstand any condition. That is, they can be seen in highly cold conditions, or they can be seen in very hot temperatures, or anywhere. So we can find them everywhere. So bacteria, they are the most abundant organism on the earth. Yes, good afternoon, guys. Mukku Komala, yes, I'm good, dear. How are you? Kasire Srivani, good afternoon, ma. Yes, today we are going to see about this bacteria. Hope you are all following me. So these bacteria are found everywhere on earth, and Anton von Leeuwenhoek first observed bacteria. Then Ehrenberg coined the term bacteria, and Louis Pasteur is the father of bacteriology because of his extensive work. And he showed that bacteria is capable of bringing significant changes in nature, and they can withstand extreme cold, heat, and drought conditions. Okay. Now coming to the morphology. Do you all know what is morphology? Yes, how they look and how they appear. Morphology. Morphology. Coming to size, it is very small. That is barely visible in the, even under light microscope. So even under light microscope, they are barely visible. Yes, this Komala, I'm just going forward with the same type of bacteria and how many types are there. In this today's session, we'll cover everything about bacteria. See, in an easy way, you can remember all the details of bacteria. So, bacteria, they are very small in coming to size. This is very small. They are barely visible under light microscope. Even under light microscope, we are unable to see that. And size varies according to the species, and majority are in the range of 2.0 μm in length and 0.5 to 1.0 μm in breadth. So you can just imagine when we are unable to see under light microscope only, then how it is highly impossible for us to see through naked eye. So the size is very small. Then next, coming to the shape. What is the shape now? The shape is how we are getting the shape. Any organism is getting the shape. The shape is rigid cell wall determines the shape of the bacteria. Now first coming to the shape, because of the presence of cell wall, the bacteria are getting different shapes. So what are the different shapes we are saying? Here we have first is spherical shape. Spherical shape means they are circular in shape. They are circular in shape. They are spherical, and what we are calling them as cocci. So the name cocci is given to the spherical shape. Then if they are elongated like rods, then it is bacilli. Then we have helical rods, which is spirillum. Okay. Helical rods are there, which is called as spirillum. Then if they are in comma shape, we are calling it as vibrion. And other than the spherical means cocci, elongated is bacilli, helical is spirillum, and comma shaped is vibrion. Other than these, all there are few bacteria that they have a capability of changing their shapes depending on their environment and the nutrients available. Such bacteria we are calling it them as pleomorphic. Example, we can say it as acetobacter. Clear? Then these bacteria, they may occur individually or in groups. Okay. That is, then they may occur individually or they occur in groups. So these bacteria, it is not mandatory that they should occur individually. Even in groups, they may occur. Now, based on whether they are growing individually or they are growing in groups, these cocci and bacillus are of different types. First, we are going to see about cocci. If only a single bacterial cell is growing, then we are saying it as monococcus. Then name only we can say, see, mono means one, so only one cell is there. Diplococcus, with name only you can say two are there. Yes, rod shaped is bacillus, elongated rod like shapes are bacillus, and circular shaped are coccus. Monococcus means single cell is there. Diplococcus, a pair of cells are there. And then I will show you the diagrams also, so that then it will be clear for you guys. Elongated structure itself is a rod shape, ma. See here. Rod, it is elongated structure. Rod is nothing but when you are elongating it, it becomes rod shape. So elongated shape is rod shape is the same, we say to the same shape. So diplococcus is a pair of cells, circular cells. If they are in forming a pair, it is diplococcus. And tetracocci is group of 4. I should know you first. Just look at this topic now. Then I'll just reply to all your questions, ma. Diplococcus, a pair of cells are there. Then we have tetracocci. With the name you can say tetra means 4. So group of 4 cells are there. Group of 4 cells are there, tetracocci. Then we have streptococcus. Streptococcus means it is a linear chain of cells in single row. So they are lying linearly with each other in a single row. Then we have staphylococcus. Staphylococcus means the cocci shaped bacterial cells are irregularly arranged. They do not follow a particular pattern. Then we have sarcinae. What do you mean by sarcinae? The cells are arranged in the cubes of 8. Right? Cocci, how many forms we have? Monococcus, single cell. Diplococcus, pair of cells. Tetracocci, group of 4 cells. Streptococcus, linear chain of cells in single row. Staphylococcus, irregular pattern in bunches. Then we have Sarcinae, which cells are arranged in cubes of 8. Then comes bacilli. We have bacilli also similarly. Monobacilli is nothing but single elongated rod shaped. Okay, now it is clear. Bacilli is nothing but rod shaped. So single elongated rod shaped. I'll mention here once again for you all. It is single elongated rod shaped cell. Diplococci is nothing but Diplobacilli is nothing but pair cells of bacilli. Two cells of bacilli rod shaped cells are there. Then we have streptobacilli. Streptobacilli is nothing but chains of bacilli appearing like a straw. So chain of, yes Mars, I know. I'll just tell you about it, all the details. First, let me just complete this session because I need to complete this topic within 30 minutes. I'll just reply you. Then we have spiral forms, right? We have spiral forms. What do you mean by spiral form? Then we have vibroid, in which cells have less than one complete twist. So this vibroid means the cells which are having only one less than one complete twist, and so spiralum is nothing but the cells have more than one complete complete twist, and it is giving distant helical shape. So monococcus, diplococcus, then monobacilli, diplobacilli. See, for your easy convenience, you can just see this diagram bacterial shape. See here. Spheres are there, see. This is diplococci, streptococcus pneumoniae. See, streptococci is there, staphylococci. Then sarcinae in the form of cube to 8 are arranged. Here tetrad means 4 are arranged. See, rod shape, nothing but bacilli are elongated, rod shaped one. Chain of bacilli. Then we have flagellated rods. Then spiral, this is vibroid. It is not giving complete one twist is also not there. But here in spirala, here one twist, one more twist, two twists are there. And here more twists are there. These are spirochetes. So with this diagrammatic representation, you can easily get it. Okay. This was all about the shapes. Now coming to cell structure. What do you mean by cell structure? So here coming to bacteria, regarding the cell structure, there are two variant components present on the bacterial cell. The first one is flagella. What do you mean by flagella? Flagella. Then some species of bacteria have flagella. And what they do, they show rapid wave-like movement. We can easily understand that with flagella, presence of flagella, nothing but indicates the movement of the cell. So some species have flagella and they show rapid wave-like movement. What they show, these cells who are having flagella, they show very fast, rapid wave-like movements. Then motility enables bacteria. What is the use of this movement or motility? This motility enables bacteria to move towards favorable environment. Whenever there is unfavorable conditions, what does happen? This bacteria moves from there and it goes to a favorable condition. So then this flagella is very essential in bacteria, which is not shown by all bacteria. Only some species have flagella, and the ones which are having these flagella, they show rapid wave-like movement. And motility enables bacteria to move towards favorable environment. Now, based on this flagellar arrangement, there are 4 types of bacteria. Based on the flagellar arrangement, how is the flagella is arranged and all? That first one is monotrichous. The name we are saying that mono means one. So in this single polar flagellum is here. See this. This is monotrichous. At the one corner or one side, single polar flagellum is there. Then second one is amphitrichous. What do you mean by amphitrichous? Amphitrichous, single flagella at each end. See here, amphitrichous. Both the sides we have one flagella. Then we have lophotrichous. Lophotrichous means 2 or more flagella, but at only one pole. So at only one pole, two or more flagella are present. So we are calling it as lophotrichus. Then we have peritrichus. So flagella is present over the entire cell, anywhere, complete on the cell wall. You can see the flagella. See in this diagram, you are able to see here. This is monotrichus, this is amphitrichus, this is lophotrichus, and this is peritrichus. So based on the flagellar arrangement, how many types are there? 4 types: monotrichus, amphitrichus, lophotrichus, and peritrichus. Monotrichus means single polar flagellum. Amphitrichus is single flagella at both the ends, 2 ends. Lophotrichus, 2 or more flagella at one pole, and peritrichus is flagella over the entire cell. Now second component of the cell wall is plasmids. Plasmids, nothing but they are small circular double-stranded DNA molecules. These plasmids are small circular double-stranded DNA molecules, and they contain fewer genes than genophore. So they contain fewer genes, very few genes than genophore are present in plasmids. Then these plasmids, they may be gained or lost without harming bacterial cell. Without harming bacterial cell, these plasmids can be gained or they can be lost. Then they can be readily manipulated. They can be readily manipulated, easily manipulated in the laboratory. Then we have these plasmids can be transferred from one bacteria to another bacteria. So by various methods, these plasmids can be transferred from one bacteria to the other bacteria. Okay. Then they are being used as agents or vectors in modern genetic engineering. They are being used in modern genetic engineering techniques. Clear, the plasmids other than genophores and all, few bacteria, they have small circular double-stranded DNA molecules. What they do? They contain fewer genes than genophore. They may be gained or lost without harming bacterial cell, and they can be readily manipulated in the laboratory. And they can be transferred from one bacteria to another bacterial cell, and they are being used as agents or vectors in modern genetic engineering. Coming to the next one, which is nutrition about bacteria. Nutrition. What do you mean by nutrition? So in bacteria, the nutritional types is based on its sources of carbon and energy. That is from where it is getting carbon and from where it is getting its energy. Based on these, that is sources of carbon and energy, the bacteria are divided based on their nutritional type. So there are 4 major nutritional groups. Okay. Then 4 major nutritional groups is, the first one is photoautotrophs. So what do you mean by photoautotrophs? Photoautotrophs. They are photosynthetic. Photoautotrophs, how the plants are photosynthetic, these bacteria are also photosynthetic. What they do? They capture light energy and transform it into chemical energy. So what they are doing? They are capturing light from sunlight and they are transforming it into chemical energy. And from where they are getting carbon? The carbon is being obtained from atmospheric carbon dioxide. And examples are chromatium and chlorobium. Photoautotrophs, what they are? They are photosynthetic and they capture light energy and transform into chemical energy. And they obtain carbon from atmospheric carbon dioxide. Both are naturally. Photo means they are getting naturally both light as well as carbon and energy. Light energy is they are converting into chemical energy, and carbon they are availing from atmospheric carbon dioxide. So they are called as photoautotrophs, and examples are chromatium and chlorobium. The next second type based on nutrition is chemoautotrophs. They are unusual nutritional adaptation. They show very unusual nutritional adaptation. They does not require sunlight and organic nutrients. So they are very unusual nutritional adaptations. They do not require both sunlight as well as organic nutrients. And how they are getting their energy? They are deriving energy from oxidation of inorganic substances. They are deriving energy from oxidation of inorganic substances, and carbon which they are getting from carbon dioxide. Example is nitrosomonas, nitrobacter, vegotova and methanogens. What are the examples? Nitrosomonas, nitrobacter, vegotova and methanogens, and these play an important role in recycling inorganic nutrients. Yes, chemoautotrophs are nothing but they are very unusual nutritional adaptations in which they do not require sunlight and organic nutrients, but they derive energy from oxidation of inorganic substances, and they are getting carbon from carbon dioxide. Examples are nitrosomonas, nitrobacter, vegotova and methanogens, and they play important role in recycling inorganic nutrients. Okay, the first one was photoautotrophs, second one was chemoautotrophs. Now coming to the third one. The third one is photoheterotrophs. What do you mean by photo? They obtain energy from light, and carbon is derived from from organic substance. They obtain energy from light, but carbon is derived from organic sources. Example is rhodospirillum and rhodopseudomonas. In photoheterotrophs, they obtain energy from light, and carbon is derived from organic sources. Example what we have is rhodospirillum and rhodopseudomonas. Then finally we have fourth type, which is chemoheterotrophs. Then they derive both carbon and energy from organic sources. What they are doing? This chemoheterotrophs, they are deriving both carbon and energy from organic sources. Chemoheterotrophs, they are deriving both carbon and energy from organic sources. And what are the two types? The two types again under chemoheterotrophs we have two types. First one is saprophytes. Saprophytes, they are free living and they feed on organic detritus. They live on, they are free living, and where they are, what they are feeding on, they feed on organic detritus from dead organisms. Example is bacillus species. So chemoheterotrophs, they are again divided into two types. The first one is saprophytes. What are saprophytes? They are free living. They feed on organic detritus and they feed on organic detritus from dead organisms. Example is bacillus species. Then we have parasites. Parasite, they derive nutrition from cell or tissue of host. So they derive nutrients from the cells or from the tissue of the host. Example is Xanthomonas, Salmonella. Okay, examples are Xanthomonas, Salmonella. So how many types are the chemoheterotrophs? Chemoheterotrophs are again of two types. That is what is chemoheterotrophs exactly? Both carbon and energy from organic sources they are getting. And they are again of two types. Saprophytes, first one is saprophytes, which is free living. That is, they feed on organic detritus from dead organism. Example is bacillus species. Then we have parasites. They derive nutrients from cells or tissue of host. That means they live in symbiotic association with the host. So example is Xanthomonas and Salmonella. And coming to the reproduction, the major commonly they divide by binary fission. Commonly they divide by binary fission, and sexual reproduction. There are three types of sexual reproduction, which is conjugation, transformation, and transduction. So there are three different processes, which is conjugation, which is transformation, and finally transduction is there. With these three processes, sexual reproduction takes place in bacteria. Commonly it is by binary fission, and sexual reproduction is by conjugation, transformation, and transduction. Clear all the details about our bacteria. First, we have seen about the introduction. That is, it is the widely growing organ. Excuse me. It is a and who has first observed this? Anton von Leeuwenhoek first observed this bacteria, and Ehrenberg coined the term as bacteria. Then Louis Pasteur is considered as father of bacteriology due to his extensive work, and he showed bacteria capable of bringing significant changes in nature. Then he can withstand extreme. They can withstand extreme cold, heat, and drought conditions. Then we have morphology. Morphology. First was the size. What was the size of that? They are very small and they are barely visible under light microscope. And size varies according to the species. And majority of bacteria range in 2.0 to 5.0 μm in length and then 0.5 to 1.0 μm in breadth. Then coming to the shape, we said that because of the rigid cell wall, we can determine the shape of the bacteria. The first one is spherical shape is nothing but cocci. Elongated rods are bacilli, rod shaped. Then helical rods are spirillum. Then we have comma shaped vibrion. Then we have pleomorphic bacteria. The bacteria which change shape depending on environment and available nutrients. Example was acetobacteria. Then they may occur individually or they may occur in groups. Then cocci or bacillus are of different types. Monococcus means single cell. Diplococcus a pair of cells and tetracocci group of 4 cells. Streptococcus the linear chain of cells in single row. Then we have Staphylococcus irregular pattern in bunches. Then we have Sarcinae cells arranged in cubes of 8. Then similarly we have bacilli forms. Monobacilli is single elongated rod shaped cell. Diplobacilli is paired cells of bacilli. Streptococcus bacilli is chains of bacilli appearing like straw. Then we have spiral forms. Vibroid cells have less than one complete twist and spirillum cells have more than one complete twist and it gives distinct helical shape. See here. This is the diagrammatic representation. Diplococci, streptococci, tetracocci, staphylococci and sarcinae. Then we have rod-shaped chain of bacilli, flagellated rods and spore form. Then spirals, vibroids, then spirilla, spirochetes. And then we have cell structure. The cell structure, here we are going to show about two variant components, flagella and plasmids. Flagella, some species of bacteria have flagella and the one which are having flagella, they show rapid wave-like movement. Then comes, based on flagella, again 4 types are there, monotrichous, single polar flagellum, amphitrichous, 2 flagella is each end, lophotrichous, 2 or more flagella at one end, and peritrichous, we said that flagella is present over entire body. Then we have plasmids. It is small circular double-stranded DNA molecule and it contains and it contains fewer genes than genophore. Plasmids can be gained or lost without harming bacterial cells and they can be easily manipulated in laboratory. And these plasmids are easily transferred from one bacteria to another bacteria and they are used as agents or vectors in modern genetic engineering. Coming to the nutrition, based on sources of carbon and energy, 4 major nutritional groups are there. First is photoautotrophs. That means they are photosynthetic. They capture light energy and transfer them into chemical energy. Example is chromatin and chlorobium. Then the second one is chemoautotrophs. It is very unusual nutritional adaptation. It does not require sunlight and organic nutrients. They derive energy from oxidation of inorganic substances. Examples are nitrosomonas, nitrobacter, bacteria, and methanogens. And these chemoautotrophs play an important role in recycling inorganic nutrients. Then we have photoheterotrophs. They obtain energy from light, carbon derived from organic sources. Example is rhodospirillum and rhodopseudomonas. Then finally, fourth one we have chemoheterotrophs. They derive both carbon and energy from organic sources. Again, these chemoheterotrophs are of two types. Saprophytes are free-living, feed on organic detritus from dead organism. Example is Bacillus species. Then we have parasites. and derive nutrients from cell or tissue of host. Example is Xanthomonas and Salmonella. Then reproduction commonly it is by binary fission. Sexual reproduction is of three types: conjugation, transformation, and transduction. Hope you all guys understood this topic. Meet you all with next topic in the next video. So do subscribe to our Unacademy channel. But before just ending this session, I would like to remind you about our Unacademy Plus subscription, which is very beneficial for all the aspirants who are willing to crack EAMCET. It is the premium content at very affordable prices with structured live courses and dedicated live doubt clearing sessions. Then we have quizzes at regular intervals, and batches are there for complete preparations, test series for DPPs, full length mocks, etc. with analysis. Then we have 12-month plan, 6 months plan, even we have 1 month trial plan. So don't miss this opportunity. Do subscribe to our Unacademy Plus subscription, and you can use my code HUMERALIVE so that you can avail extra discount. So see you guys. Meet you in next session. Take care. Goodbye.